Yes, and welcome everyone in today's session of the BioClock Academy series. It's nice to see some of the regulars again, but also some new faces. My name is Hannah Brooks, and together with Laura Pape, we will host today's session. We are both PhD candidates from the BioClock Consortium. That's the organization that organizes these seminar sessions. So in short, the BioClock Consortium is a Dutch organization that researches the biological clock in different contexts. So human health, society, the environment, all of that. And with this seminar series, we actually want to equip you with some basic concepts, but also knowledge straight from some experts in the field who are working on very different subjects within the field of chronobiology. So these sessions are held every third Wednesday of the month. For the timing of today, we will get 40 minutes talk approximately, and then there's, after the recording, 15 minutes for discussion, so we can end about at 5 p.m. At European time. Something else, well, you can already put your questions in the chat during the talk, so we can then come back to them afterwards, but you can also just, once the recording is finished, ask them via the microphone. And I think that was all the practical information, so then I give the floor to you, Laura, to introduce our speaker of today.
Yes, thank you, Hannah. Yeah, so today we have the pleasure to have among ourselves Dr. Andrew Beale. He's a senior scientist at MRC Laboratory of Molecular Biology in Cambridge in the O'Neill Group. And he's interested in the interaction between environment, circadian biology, and health. Specifically, he investigates the cellular mechanism of circadian rhythms in nucleate and anucleate cells. Correct me if I'm wrong, but yeah, warm welcome to you, Andrew, and we're looking forward to your talk.
Thank you. All right, well, thanks for the introduction. As Laura said, I'm Andrew, I'm from the UK, and it's a great pleasure to give this international talk via Zoom. I've not done one before, and I'm really, really grateful to be able to have the opportunity to talk about my research and something that I'm interested in, which is comparative molecular chronobiology. And I'll describe what that means in a bit. But first of all, I just wanted to start off by describing this image, which I put, this piece of art. Earlier in the year, I gave a talk at Pint of Science, which is a kind of public engagement event that runs in pubs and bars throughout the world. And scientists are invited to give public-facing talks. And as part of that process, in Cambridge, they paired us with some artists. That could be a musician, or it could be a fine artist like I did with Caro Leopold. And Caro decided to do a drawing based on my talk, basically the description I gave. So kind of what Laura just introduced, that amount of detail. And what she picked out from it were the two most favorite things I had about the talk. Firstly, this idea of the Linnaean clock of flowers, which was invented by Carl Linnaeus, and describes how over the time of a day, that over a whole day, flowers will open and close at different times. And he proposed that you could generate a clock based on which flowers are open at any time. Still hasn't been made, so it's a challenge that's still open to anyone with a keen interest in gardening. But the other aspect of my talk was the idea of animal biology and how animals compartmentalize their activity to the day and the night. And so she picked out a few different animals, bats and birds and moths, that do things at different times of day. And that's something I will talk about a bit later. But I first wanted to start off with this quote, which is, "Nothing in biology makes sense except in the light of evolution," by Theodosius Dobzhansky. It's actually the title of an interesting and philosophical essay. That's not really why I chose it, although you can go and read it if you like. I actually chose it because it's famous as describing a view of biology where biological phenomena can only really truly be understood through the lens of evolution. Now, the type and magnification, if you like, of your lens can take many forms, but we can describe two as ultimate and proximate explanations, which deal with fitness and selection and mechanism, and I put that in quote marks deliberately. So, for example, colourful birds, an ultimate explanation of their colour could be that brighter plumage results in attracting more mates, more chances to reproduce, whereas a proximate explanation might deal with the cellular mechanism by which the colour is laid down in the feathers. Now, we can apply those two terms to chronobiology and simply put that could be why have a clock and how does it work? But ultimate and proximate explanations are actually intertwined entities and they can't completely be isolated from one another. But today I'll describe two examples from my own career, my own work, that focus more on one than the other. And that is the story of circadian cavefish and the cellular origin of mammalian diurnality. Now, I'm not the first person to look at chronobiology in this way, and I could list dozens of giants' names upon which I stand. Perhaps the most significant paper that's coloured my research has been this classic from Colin Pittendrigh from 1993, who discusses the field of chronobiology and his own work from his perspective as an evolutionary biologist. And in particular, two phrases from it which lend themselves to this thinking of ultimate and proximate explanations in chronobiology, and they are the day outside and the day within. Now, the language of the day outside may be familiar from the opening lines of many chronobiology papers, which describe the rotation of the Earth. So here, Pittendrigh writes, from life's outset, the major environmental cycles of day, tide, month, and year have confronted natural selection with windows of opportunity and hazard that recur with precisely predictable frequency. Now, that perspective might have been pondered by Darwin himself, as Tiago Gianjardi and I explore in this recent perspective just out a few months ago. The day within, however, this deals with the importance of temporal order within the organism, within the cell, separating metabolically incompatible processes in time, like photosynthesis and nitrogen fixation. And what Pittendrigh writes about this is that such selection by temporal order internal to the cell is surely as old as that exerted more directly by the external cycles themselves. Now, these are kind of ultimate causation questions, which suggest there's a difference between extrinsic or day outside explanations and intrinsic and day within, and the evolutionary value of those two things in respect to the circadian clock. But what do I mean by this? Well, first of all, I really recommend Louisa Jabert's review, which really looks at this idea of adaptive fitness question in detail and really critically assesses the evidence. And one of the approaches they describe in that paper is an experimental approach to determine whether a clock has intrinsic value from the day within. And they run competition experiments under constant conditions. Now, if the important thing about a clock is its extrinsic value and its relationship to the day outside, in conditions where the selective pressure is absent, so constant conditions, in this example, it's constant light, you would expect that 50-50 mixture of a clockless and a clock-containing cell to remain the same. There's no advantage to having a clock. It wouldn't outcompete the clockless strain. However, if the important thing is its intrinsic value, the internal temporal programming or the separation of photosynthesis and nitrogen fixation that I mentioned before, having a clock might continue to be of adaptive value to an organism, even in constant environments. And so in that case, it would outcompete the mutant strain, and the mutant strain would drop to very low values or amounts in the population. Now, this is a nice experiment and very clear, but are there any systems we can already use to answer this question? Well, one way is to examine organisms that live in constant environments, such as caves. Caves have been described as natural experiments to investigate environment-phenotype relationships, and are especially useful in determining a feature's relationship to light. So cave organisms like these share many common features, such as being blind and albino, and therefore demonstrate the extrinsic value of pigment and eyes. They are no longer of adaptive value in the darkness and have regressed. Now, in the early days of chronobiology, they used to research many different organisms, lots of organisms, including organisms in caves. And in 2 papers from the 60s and 80s, they would get behavior-based readouts like this. In these 2 papers, they don't detect any reliable circadian rhythm under constant conditions, either in the cave crayfish or in this particular species of cave fish. There is no obvious rhythmicity at the behavior level in those organisms. And by that, by their existence, that agrees with possibility 1, that the clock is only of extrinsic value and the day outside is the most important thing. Now, I've made an assumption here, and they have too, that those cave organisms, that doesn't mean it doesn't completely equate with that experimental example from earlier. And that assumption is that the original forms of those cave dwellers had eyes and pigment and behavior rhythms. But it's possible that they didn't. We don't have that wild type or control group. So during my PhD, we sought to explore this intrinsic and extrinsic question again, using this model, Astyanax mexicanus cavefish. Now, Astyanax mexicanus is an absolutely amazing model for working in the fields of developmental biology and circadian biology, because as well as having multiple different independent populations of cave fishes, so showing convergent evolution at the level of the cave, their ancestor, or something very similar to it, exists in the rivers that surround those caves in this region of northern Mexico. And so if you can compare cave fish and surface fish in Astyanax mexicanus under different conditions, you can do that wild type versus knockout experiment. And so we did this, and in modern lab conditions, again, compared to the control group now, which is the surface fish, which shows rhythms under constant conditions, the cave fishes don't. Now, that agrees with those 8 papers from the 60s and 80s and so on, and suggests that it's extrinsic value, the day outside, that's important. But the readout in this example is behavior-based. And I just want to take a moment to say that activity might be the wrong thing to look at in this case, because it can be problematic. Because in many cases in nature, behavior is naturally arrhythmic. And to illustrate that, we can look at the case of cathamerality, which is a case of, or a natural case of arrhythmic behavior that's often context dependent. For example, reindeer in northern Norway and Svalbard undergo periods of complete absence of behavior rhythms during the constant dark of the winter months. Wild boar in the border of China and Russia also experience this arrhythmic behavior type phenotype in that location, whereas elsewhere in the world, they have very strong nocturnal or diurnal patterns. And so cathamerality does demonstrate the importance of the day outside in maintaining rhythmicity, but the absence of rhythmicity doesn't necessarily rule out the importance of the day within, that intrinsic value. And so this is where we turn to looking at other markers of circadian rhythmicity and that molecular chronobiology I mentioned in the introduction. So you might all know that the circadian clock is a cellular and molecular entity with oscillating mRNA abundance, such as described as the TTFL model, as well as various pathway activity rhythms, for example, rhythmic protein synthesis from the ribosome, or even post-translational modifications like phosphorylation or other aspects of metabolism, for example. But oscillations of these core genes, such as the pers and cries, which is shown in this graph here, serve as very strong markers of a functional molecular clock. And so we sought to look at the molecular chronobiology of the cavefish to answer our evolutionary question. However, back in the late 2000s and early 2010s, with cavefish, it took a lot more effort than it does now to look at molecular chronobiology, as we didn't have a transcriptome or a genome to allow us with the available techniques to easily measure the expression of these genes. So we had to do it the old and slow way and clone out these genes one by one from cavefish by degenerate PCR. And then, and only then, could we examine their expression by qPCR in serial samples. And so we did this in sample populations of surface and cavefish under light dark conditions and constant dark conditions. And what we saw is, again, the surface fish have a strong rhythm in its molecular clock, as shown in red, but the cavefish also do have a rhythm in the molecular clock, shown in blue and green. Although the actual pattern has differed, it's still there. Now, bringing it to the 2020s, other people have taken on this work, including under the strong leadership of Alex Keane. And in this paper from 2021, they used RNA-seq to examine multiple species of cavefish, or multiple populations, Merlino, Passion, and Tinaja. And what they found was very similar to our paper in 2013, that there is a pattern of rhythmic expression of these genes in cavefish. It's just altered. It's the repeated evolution of a disrupted circadian oscillation. And so what that means is, within the context of cavefish, both the day outside and the day within are important. There's a reduced rhythmicity in an arrhythmic environment of behavior and of the molecular clock, but there isn't a complete loss, indicating some kind of functional constraint or importance of the clock, even under constant conditions. Now, when working in the space of comparative biology, especially molecular chronobiology, methodology is really critical. And Louise's Jabert's review makes that super clear. Now, I mentioned that reindeer have periods of cathemerality or arrhythmic behavior, depending on the season. In 2010, this paper came out that looked at the molecular chronobiology of the reindeer using the expression of this gene, one of the clock genes, BMAL1. And from the absence of any rhythms under constant conditions, they made this conclusion that circadian clock is not required in an Arctic mammal, which matches that behavior arrhythmicity. But just a few weeks or even a couple of months ago, another paper came out in JBR that had the complete opposite conclusion, in that there are rhythms in the reindeer under constant conditions, but they are altered, just like we saw in cavefish. Again, kind of demonstrating that the difference in methodology between this paper and this paper has given them two completely different answers. So check your methodology. Now, let's change gear a bit and move on to my second example, which is the origin of diurnality in mammals, where I'm actually focusing on the proximate cause to this question. Now, it's commonly thought that the ancestors of modern mammals were nocturnal until the extinction of the dinosaurs at the Cretaceous-Paleogene boundary facilitated a rapid expansion into the diurnal niche. This is described as the nocturnal bottleneck, where early mammals were confined to this nocturnal niche until the dinosaurs disappeared. Today, diurnality is present in multiple mammalian families, having arisen independently multiple times. But given how important and how critical diurnality is part of our human evolutionary history, the mechanism facilitating that switch is relatively understudied. Now, in the studies that have compared them directly, this master timekeeper, the SCN, functions relatively similarly in diurnal and nocturnal mammals, receiving light input directly from the eyes to generate an internal representation of solar time. However, unlike the SCN, the major behavioral and physiological daily rhythms in mammals, as well as per oscillations, the molecular clock in peripheral cells, are oppositely organized between nocturnal and diurnal in the mammals, and instead coincide with their activity pattern rather than solar time. Now, this organization suggests a switch downstream of the SCN, as we know how the SCN signals to the rest of the body, perhaps some kind of signal inverter, which takes the signal from the SCN, which ancestrally imparted nocturnal physiology, and inverts it to impart diurnal physiology. And we considered whether a cell intrinsic mechanism could account for this. However, at the cellular level, evidence suggests that the major hormonal cues, such as insulin and growth factors or glucocorticoid signaling, acts relatively similar between mice and men, setting them to the same phase, if you like. But what about temperature, this non-hormonal cue? Well, although generally considered subservient to hormonal cues, physiological rhythms in temperature are sufficient to synchronize cellular clocks throughout the body and brain. Whether temperature-mediated timing cues act similarly on cells from diurnal and nocturnal mammals has not been tested, so we explored this in vitro using programmed cycling incubators of temperature while simultaneously monitoring the activity of the circadian clock via luciferase reporters such as BMAL1 luciferase, as I mentioned in that reindeer paper. And amazingly, primary fibroblasts from multiple mice and multiple humans consistently entrained oppositely to these very same temperature cycles. So this line drawn on the x-axis here just illustrates that they peak and trough at opposite times of day. And the circadian plot on the graph just plots the peak activity of the reporter with respect to the last transition to 37, showing that they entrain in antiphase to one another. Now, that opposite entrainment I observed for other cell lines, different tissues of origin, as well as different reporters, as well as smaller magnitude temperature cycles, which, although they changed the absolute phase of entrainment, as would agree from much of entrainment theory, it didn't change the phase difference between the nocturnal mice and diurnal humans being still around 10 to 12 hours apart. Now, obviously, there's much different between mice and men, so we were curious to see how this entrainment difference spanned across the mammalian family. And thanks to collaborators, I was able to create primary fibroblast reporter lines derived from more diurnal and nocturnal species, from large sheep to little striped mice. And I repeated the very same experiment. And rather amazingly, cells from these other diurnal and nocturnal mammals grouped together and 12 hours or so apart from one another. And so with these lines, which are of course isolated from the brains and cultured in a dish, we find our signal inverter, the conserved differential response of the cellular clock to this temperature stimulus. Now, just to make clear here that we can't infer too much from the phasing of these cells in these in vitro experiments to how they would behave in vivo. There are myriad other cues that cells and tissues integrate in vivo in an organism to achieve that stable phase of entrainment. But what we have done is show that however diurnality is achieved, its mechanism is encoded within the cell, which allows us to drill down to its molecular basis. Now, the inverter is not some specific single clock protein alteration and is not due to the differential response of the heat or cold shock pathways. It's a global difference of the cellular states of nocturnal and diurnal mammals. And to illustrate what I mean by this, we looked at the total phosphoproteome of mouse and human cells under different temperature conditions, such as constant high or constant low temperature, here being 32 degrees, or in response to a step up, an increase in temperature, or a step down, a decrease in temperature. Now, globally, the phosphoproteome responds differently in those two species. Under constant temperature differences, the medium phosphorylation state of humans is increased at higher temperatures, whereas it's increased at lower temperatures in mice. And in response to a temperature step, the humans react to increased phosphorylation upon a temperature increase, whereas mice, on average, react the opposite, reacting to increased phosphorylation upon temperature decreases.
Now, we can go further down this proximate cause and mechanism route by identifying the drivers of this response, which will be mainly the kinases causing this response. And we can do that either by looking at the matching phosphosites between these two populations and seeing how they are annotated according to databases, or we can take a different approach and see whether the characteristics of the target phosphosites tell us anything about which kinases phosphorylate them. This is what I mean by phosphoprotein motif analysis, which looks at the amino acid sequence surrounding those phosphosites to allow identification of the kinases responsible. And if you're interested in this kind of analysis, I really recommend this paper from Johnson et al. In Nature just last year, which really tells you something about this method. Now, we were interested really in which kinases could be driving the opposite response. And so we looked for common motifs in these two opposite populations. And in those two opposite populations, we identified an enrichment for basic amino acids, so these arginines and lysines, meaning that these phosphosites that are responding to temperature oppositely are being targeted by kinases that are basophilic. They like this basic sequence surrounding. Now, basic residue motifs are targeted by diverse basophilic kinases. They're actually on all of this side of this wheel, but they include the regulators and effectors of the AKT-mTOR pathway, as well as these kinases, the Wnt kinases. Now, those pathways and kinases are ubiquitous essential proteins that function as major determinants of metabolism, protein translation in the case of the TOR signaling pathway, as well as intracellular water balance and ion content in the case of the Wnt signaling pathway, and cellular state more generally. Their activities are coordinately and circadian regulated, and they've often been described as a metabolic rheostat, integrating many different metabolic and extracellular signals to maintain cellular homeostasis. And as such fundamental proteins, they are attractive candidates to mediate a global response to temperature. Now, they're also sensitive to another cellular state, that of intracellular water potential. Now, a response to water potential isn't something that's fully apparent from pathway diagrams like these, but it is a bit clearer when we consider the intracellular environment with the help of these amazing images from David Goodsell. And those images really emphasize how busy a cell is. On the left, for example, is protein synthesis in action in the context in which you would find it in the cell, with the ribosomes in this blue-green, the mRNA being translated in yellow, and the protein being produced in orange. All of those components require water to stay in solution and not cause problems within the cell. But as you can see, there's not a lot of space, not a lot of water going round. In fact, the cytoplasm, which is where these will be active, is super finely tuned and responsive to this availability of water, and cells react to defend it. And we've recently described some of that cellular mechanism in this paper from last year. But another thing we found in that paper is that with respect to water potential, temperature and external osmolarity actually act very similarly. And that suggests a very clean hypothesis. If Wnt and mTOR pathways are involved in sensing water potential, as has recently been described, we may be able to entrain cells of diurnal and nocturnal mammals with extracellular osmolarity. We may be able to get a differential entrainment result, just like temperature. However, doing that isn't trivial. Now, cycling temperature is relatively easy, as you can program the incubator automatically to switch between two temperatures. And indeed, that's what we set up in the lab. To get cycling osmolarity, extracellular osmolarity though, you need to be able to manipulate the culture medium precisely without triggering other response pathways, such as the growth factor signaling, which you get when you change media on cells with media containing cerium in the normal way you would do in cell culture. That isn't an easy endeavor. And so I actually had to build a whole new system and design a microfluidic system, which allows the controlled delivery of defined media automatically and programmed over time by this LSYS microfluidics manager. Like this, I can give many days of cycling media changes, taking the media from one tube or the other without handling the cells at all, and are able to entrain to cycles of basically any media composition I want. But when we do this experiment with cycling extracellular osmolarity, so hyperosmotic meaning more concentrated, and isoosmotic is the regular medium, just like temperature and exactly like we predict, human and mouse cells entrain with opposite phases. Now, interestingly, and supporting this duality hypothesis from that paper I mentioned before, the resulting phase of the two entrainment stimuli, so osmolarity and temperature, is actually almost the same, with human cells peaking during the lower osmolarity portion of the cycle and the higher temperature portions, and mouse vice versa. And indeed, I chose the magnitude of this hyperosmotic stimulus based on our mechanistic findings from that paper, where the size of that stimulus is equivalent to this amount, 5-degree temperature shift. And so we can actually now refine our approximate cause explanation that the cellular clock actually responds to changes in solvent thermodynamics or water potential. And that response depends upon the cellular state when that extracellular signal is received. Now, we can actually manipulate that cellular state using a variety of inhibitors, including those of the TOR pathway, which drastically change how a cell responds to a temperature stimulus. In this experiment, cells or tissues were cultured under chronic TOR inhibition using this INC128 molecule and exposed to the same temperature cycles for a number of days before looking at the entrain phase as the experiments I've done previously. And compared to the control conditions, which are given in black, those treated cells shift by a very big magnitude, more than 4 hours, with the nocturnal mouse cells responding more strongly than the diurnal humans. The very same response is seen in tissues taken from animals and cultured ex vivo. Large shifts in the temperature response under TOR inhibition is shown as the difference here between the treated in blue and the control condition in black. There's still one big question though. How does all of this relate to the situation in real mammals running around freely behaving? Well, interestingly, previous experimental data actually supports this idea that TOR activity and cellular state is linked to the diurnal versus nocturnal behavior and physiology. Because under more natural low net calorie conditions and therefore reduced TOR activity in altered cellular state, like the wild as described in this paper, mouse behavior and physiology becomes more diurnal than when they are fed ad libitum, when they've got plenty of food, whereas the SCN still stays as that signaler of solar time. Now, a recent paper has mapped some of this change to brain regions, including the lateral habenula, but we sought to replicate this finding or these sets of findings experimentally. Now, one way to do this would be caloric restriction, which reduces TOR activity, but as shown by the Takahashi labs, imposes a feed-fast cycle as mice self-select when they actually consume their limited food. We wanted to avoid that enforced entrainment cue, of course, to replicate a more natural diurnal-like situation. And so instead of total caloric restriction, we turned to diet modification via specific amino acid restriction. while maintaining the food ad libitum. And so restriction of TOR activity by methionine amino acid restriction has been demonstrated numerous times. And here in this study from Claire Barcena, she shows that under methionine restriction, the TOR activity, as shown by the activity of the phospho S6 kinase site, is reduced. So we fed mice ad libitum, so they had plenty of food at all times of day, using the same diet formulation, which we would predict to be able to reduce their TOR activity, for 4 weeks under an LD cycle. And what we saw was actually a small but significant shift of the mouse activity into the daytime, with minimal weight loss, as would be expected by total caloric restriction. Now, an even more extreme TOR modification without that feeding cue can be achieved by finely controlling the energy balance of mice a different way, by giving food only after a certain distance run on a wheel, a kind of reward type pathway. One of the things this method, which was developed by Roelof Hut and colleagues in the Netherlands, does is to tip animals into a status of low energy balance and almost starvation-like states, which at the cellular level switches cells from that normal state of protein synthesis to a reduced TOR activity state of autophagy, just like the TOR inhibition we'd used in vitro. Now, under those conditions, these work for food mice gradually become more and more diurnal, as shown by this daily plot here, again, agreeing with our cellular model. So our proximate cause then is a modifiable TOR and Wnt-controlled thermodynamic cellular state between diurnal and nocturnal organisms, which reveals itself under in vitro entrainment conditions, but applies in vivo under reduced energy balance. Now, zooming back out for a second from our proximate cause to an evolutionary outlook, I started out this whole section by saying that diurnality evolved independently several times. And it's therefore likely that it's acting through many genetic loci. And these have long been assumed to be different between diurnal lineages. However, if these Wnt and mTOR activity patterns are an efficient evolutionary means by which to achieve diurnal phenotypes, then convergent evolution would be able to be detected by comparative genomics. Now, to answer this question, we collaborated with Matthew Christmas and the Zoonomia project. Now, Zoonomia is a vast comparative genomics resource of more than 240 diverse mammals, and has been used to identify genomic segments that are both conserved across all species or changing with respect to some phenotype. And Matthew's paper from Science in 23 describes hibernation, for example. But we asked the question of the diurnal and nocturnal phenotype, classifying 70 of the mammals in the database as diurnal and 110 as nocturnal. Segments that say the same across most or all of the mammalian species sampled, which is roughly 100 million years of evolutionary time, likely serve some important function and hence would cause disease if changed by mutation. And indeed, what we see in these conserved regions is a large enrichment for genes involved in the adaptive immune response, which are conserved between diurnal and nocturnal mammals, indicating their very fundamental role in the behavior and physiology of those animals. However, amongst the genes that are evolving faster in diurnal than nocturnal mammals are genes involved in the olfactory pathway, which might be expected from the very different sensory challenges of day and night activity, as well as key regulator of TOR activity, RAG-B, and Wnt1 itself, in agreement with our cellular data and hypothesis. So today, hopefully, I've given you some insight into researching ultimate and proximate questions in chronobiology, using cavefish, which have clocks and at least in part suggest they are intertwined with making a day within, and that's the important thing. and mammalian diurnality, which can be explained using a cellular mechanism of differential wing and mTOR activity. I also hopefully have encouraged you to try out some non-model organisms, depending on your specific question, but also stress the importance of methodology, as well as the power of comparative molecular chronobiology to gain a deep understanding into our general sense and understanding of time in biology. So that's the end of what I've got to talk about today. I just need to end now by thanking everyone who's been involved in the research part of my talk, including everyone at the LMB and during my PhD at UCL. But part of the BioClock remit is to give a little bit of perspective on the career path, which has taken me to here. And so my next slide describes this like so on a timeline. My career path academically looks like this, starting off with an interest in chemistry, broader biology, paleontology, and finally getting a degree in natural sciences and zoology. Then went on to do a PhD at UCL on firstly developmental biology, but I rotated in a lab that does circadian biology, and that's what I ended up getting my PhD in. And I'm actually really grateful for David Whitmore, who gave me that little talk, because that has set the course for the rest of my career in circadian biology. After that, my research career followed postdocs, short postdocs, still within circadian biology, but sampling human biology for the first time at the level of the population, and then red blood cells, which finally took me to John's lab at the LMB, where I continued my mammalian molecular journey with a whole lot of catching up to do on biochemistry and proper cell biology, which I neglected during undergraduate. Now I'm a permanent member of the LMB, and I'm looking forward to many new adventures. But if you've got a keen eye on this timeline, you notice some of these dates don't add up. My PhD, for example, did not take nearly 5 to 6 years long. And in this graphic indicates periods I've had inside research science in red and outside research science in blue and green, because my pathway to now has not been linear. After my PhD, I actually decided not to continue in research. It's not that I didn't necessarily enjoy my PhD. I really enjoyed it. I just thought I might enjoy science communication better. And so I took jobs in the London Science Museum and at the Royal Institution in 2 science communication roles, which I enjoyed, but subsequently discovered it probably wasn't much for me. I lasted about a year in that pathway. But partly the reason for finishing that was that my wife's work took us to live in rural Mozambique for 3 years, where I wasn't really a scientist. I was a bit of a jack of all trades, to use an English term, using my skills in just general communication and computers to train her colleagues on computers and using them, playing with my Mozambican neighbours' children and learning Portuguese, as well as learning about the hygiene and sanitation work that she was doing. At the end of that time, then I moved back into research, and that was only thanks to the UK Clock Club having free meetings, because one of the trips back into the UK, I went to one for free, and there I met Malcolm, and that's the start back of my research career. You'll also notice these gaps here where, of course, the lockdown interspersed these periods inside and outside research, particularly if you have children like I did. And then I went on to have a significant time of parental leave with my youngest child. So it's only really since the end of 2021 that I've had a solid chunk of postdoc time really to develop these kind of skills. And I still kind of maintain that this random career path is actually not necessarily the way you would tell it on a CV, but has definitely benefited my understanding and love for research science. And so for those reasons, I've got a whole second page of acknowledgements, which include, of course, the research side of people who I mentioned, David at UCL, but also my undergraduate supervisor, Marana, at Cambridge, the people from my time in Surrey, the clock community in the UK, as well as internationally, and especially Maria de la Paz Fernandez, who worked alongside me at SRBR, but also the communications part of my career, and then the various people who've coloured my outlook, I would say, on research, but who don't necessarily work in research themselves. So with that, I'd like to finish, and thank you for listening.
Thank you very much, Andrew, for your very nice talk and also for this really nice overview of your career path.